Welcome everyone to another session of Geeky Latinas. We are so happy to have you with us. I am Junisa Ann, a Geeky Latina volunteer. For those who don't know, Geeky Latina is an educational platform that encourages young people to become professional in STEM's career, which are science, technology, engineering, and mathematics. Today, we have an interesting session. But before, we have to go over our rule for this session. Number one, check camera connection. Check connection, camera, microphone, router, and any other hardware to be used for video conferencing. Good lighting. Try to have good lighting, either with natural or artificial light. Identify yourself. Remember to rename yourself on the Zoom platform, indicating name and surname, country of residence. Example, Maria Torres, Mexico. Choosing an appropriate background. Choose a simple background, avoiding any bright, exotic, messy, dirty background. Choosing an appropriate wardrobe. Dressing correctly and completely is important because any movement out of the frame can reveal something that should not have been seen. Have a good framing. We must look for a good framing with a certain margin of movement so that we can be seen correctly. Number 7, avoidance of ambient noise. It is very annoying to hear noises of all kinds when someone is talking. Bricks, banging, music, discussion, television, or radio in the background. Start on time. Be on time so that the meeting lasts the scheduled time. Thank you all for your attention. Today, our speaker for today is Alicia Villoyaputi. Who is Alicia Villoyaputi? Alicia Villoyaputi was born in Maracaibo, Venezuela, and moved to Canada in 1996, making it her home. She currently lives in the city of Guelph in Ontario, the largest English-speaking province in the country. Alicia obtained a master in immunology from the Venezuelan Institute for Scientific Research and a PhD in cellular and molecular biology from the University of Toronto. Following a postdoctoral fellowship at the Lunenfeld Tannenbaum Research Institute in Toronto, Dr. Villoyaputi joined the University of Guelph, where she currently is an associate professor at the Department of Biomedical Sciences. Alicia's research focuses on understanding how cancer disseminates the distant organs with the aim of improving early diagnostics and treatment of metastatic disease in breast and bone cancer patients. She is the co-creator and co-leader of Guelph Dog Bone, a multidisciplinary research team aimed at the discovery, preclinical, and clinical testing of biomarkers and new therapies for dog and human bone cancer. Alicia is also the co-creator and co-leader of Geeky Latina, this program, which you know is a multi-ally program aligned with the UN Agenda 2030 and targeted the minority youth of the Americas, whose purpose is to enhance their interest in STEM careers, sustainable innovation, and global citizenship. Due to her contribution to cancer research and Canadian society, Alicia was named a top 10 most influential Hispanic Canadian in 2016. In her free time, Alicia enjoys reading, watching movies, and hiking with her family. She has 2 kids, 1 human and 1 cat. With the cat, Luna, she enjoys doing yoga every morning, and she is not ashamed to confess that her cat is 10 times better at it. Thank Alicia for being here with us today.
Thank you very much, Yunisha, for that amazing introduction, very, very complete. So I'm very happy to be here again and with people who already know me because I gave you the introduction to the program before. So I will be sharing my screen now. And please confirm that you see it when I share it. Are you seeing my screen now with the presentation?
Yes.
Or with the Zoom, the one that has the presentation, right?
Yes.
Correct. Okay. So I am going to start my presentation then. And you should be seeing my screen with just one slide, correct? Okay, perfect. Okay, so let's start talking about cell biology then. My presentation is entitled Thanks to the Microscope, the Daily Life of a Cell Biologist, okay? And so we'll be talking a lot about what a cell biologist does and why we use the microscope so much. But before we get into the theme, I would like to introduce myself so you can know a little bit of where I come from and where I am now. So as Junisa explained in her introduction, I was born in Maracaibo, Venezuela. And what you see here are the houses that are typical of the center of Maracaibo. It's a very, very colorful city. So we like a lot of very brilliant and very powerful colors. And what you see here is this Angel Falls, the Angel Falls, which is the typical nature hallmark of Venezuela, my country. And this is in the south of the country, in an area that is known as Canaima. And here is a picture of me when I graduated from undergrad, which is something in between a bachelor's and a licentiature and a master's here in North America. And here is when I graduated from the University of Toronto with my PhD. So as Yunisha mentioned, I actually have a number of degrees, and I highlighted here the center of my studies, the topics, the subjects, so that you can see that from my start until the end, what I was doing was narrowing and narrowing my knowledge. So I was specializing more and more. And this was very important for me because I wanted to be a scientist. So I have about 13 years of higher education, and I don't want to transmit the wrong message here and make you think that you need to have 13 years of higher education. In my case, I wanted to do it, and I wanted to have a master's and a PhD and a postdoc and all of that because I wanted to be a scientist. And to be a scientist, you need to accumulate a lot of knowledge, because what a scientist does is to propose ideas and test ideas, okay? And test ideas to address a particular problem. In my case, I am interested in addressing how cancer moves to one side where it originates to a different site in the body, and how it colonized that site to basically cause mortality, because we want to avoid this problem. And so because of that, I needed to accumulate a lot of knowledge about cells, about how your body responds to cells that are foreign to them, and that's why I study immunology. Then if I want to know about how cells behave and how molecules in the cells behave, that's what I did, cell and molecular biology. And ultimately, I did signal transduction because I wanted to know how cells transduce signal from the outside environment to the inside to respond to the environment where they are. So that's why I did this. And then because of that, I was able to become a professor and a scientist at the University of Guelph, where I am now in Ontario, Canada. And so I can say this is my academic achievement, but I do have my personal achievement, which I treasure a lot, and that is my family. And this is what you can see here. So this is my husband, Stuart, who is also a professor. He's a history professor here at Guelph. My daughter, Lucia, my daughter, Luna, who is the cat, and my spouse. So Luna and Lucia are both very interesting people because they behave very similarly. They are both very interested in my attention. The only difference is that one is have more hair than the other, really. So these are my two kids here, and also I am very happy to be in a healthy and welcoming environment and a secure environment where I live here in Canada. And to make you laugh a little bit, we have actually done a Simpson version of this picture where you can see all my family seen as the Simpsons. You know, if you have seen the Simpsons, what the Simpsons stars every time, you see this thing where they sit and watch TV. Well, this is us in the same picture making to the Simpsons. So my current environment, as I mentioned, I live in Guelph, and I work at the University of Guelph. And Guelph is about an hour Toronto, okay? So you probably know Toronto because it's a very large city and very famous worldwide. So yes, we are in the province of Ontario, which is the same province where Toronto is. And Guelph is very interesting because it's a city, but it's very small. It's about, I would say, if we count the growing area, maybe max 140,000 people now. But we are surrounded by nature, and there is a actual nature paths that you inside the city, so we can enjoy a view like this by just going away from our house for 10 minutes walking, and we get into this area and a beautiful nature place where you can even see deer sometimes. You can see some wildlife. And here are some hallmarks of Canada. You probably know the CN Tower in Toronto. Everybody knows it. And if you have traveled to Canada, you probably have gone up the CN Tower and look at Toronto from there. And this is the Niagara Falls, which are in the border with the US. And is this what you see here is the boat that is called the Maid of the Mist. And people go into that boat, and then they travel, and then they get very close to the falls and get wet. So people do that, and it's a very touristy thing to do here. And these are the symbols of Canada, or flag, the Canadian flag with the maple leaf in the middle. And this is a joke about the way Canadians talk. We say a lot of A. How is it going, eh? How's the weather, eh? So the A is something very Canadian. So if you hear somebody say A, for sure it will be Canadian. And you can see here, or moose, or beaver, maple syrup, the hockey stick, because play hockey here. And the symbols, the totems of the native people of Canada, the indigenous people. This is the hat of the typical policeman of Canada, which is called a Mountie, okay? So, and here is our university. So what you see here is one of the main buildings in the University of Guelph. And yeah, proudly showing the Canadian flag because we are Canadian. So the other thing that I didn't mention to you is that similar to the United States, we also have a Big Apple. But different from the United States, our Big Apple is not a New York, but instead it's actually a place where they sell apple pie. And this place is located in Ontario. And indeed, they have a huge apple, a big apple, where you go there and take pictures of yourself in front of the Big Apple. And it's a very famous touristy place. And this is like a big supermarket where they sell all apple things, from apple pie to apple wine. So you name it, you can find everything that you want made with apple. Okay, so that's our Big Apple. So let's now start talking about cell biology, and I want you to pay attention to this because the Kahoot questions are going to be focused on what I will be talking to you about, okay? So cell biology, a world of possibilities. What I am calling this a world of possibilities? Well, because really there are so many possibilities of things that you can do with cell biology. To begin with, everything that lives has cells, okay? So all living things from all different kingdoms, we have 6 kingdoms, okay, have cells. And because they have cells, you can study all of those organisms by knowing cell biology, okay? So you can work in zoology, which is the field that studies animals, botany, that studies plants, microbiology, that studies microbes in general, bacteriology, bacteria, virology, viruses. Now, viruses are an interesting thing. They don't have cells, but they need cells to live. They need cells to divide and to cause disease, obviously, because the disease is caused because of what they do to cells, okay? So virology is also studied by cell biologists. Mycology study fungus, parasitology, parasites. Histology is the science of the study how cells organize in tissues and then in organs so that we can function as individuals. Any animal can function because of the way things organize in tissue, plants as well. Anything that has tissue, which is an organized group of cells that has a function, anything has cells, okay? So if you are a histology, you need to know about cells, and a cell biologist can work in histology. You can work in immunology because immunology studies the immune response, and immune response is mediated by cells, okay? You can do toxicology because toxicology studies how toxins damage cells, okay? And how cells respond to toxins by developing mechanisms that actually destroy them inside them. So that, cells again. And finally, hematology, which is the science that study the blood and its components. And so study how the blood forms and what is typical of normal blood and what is typical of abnormal blood so that we can diagnose disease that is related with changes and abnormalities in the blood. So you can basically work on all of these fields. You can also work in applied fields that are more toward a particular goal in, let's say, biomedicine, for example. What is biomedicine? Biomedicine is what I do. Biomedicine is applying cell biology knowledge, in my case, to the understanding, could be the diagnosis, could be the treatment of a given disease, even the prevention of that disease. Biotechnology, what is biotechnology? Well, when you, in the case of cell biology, you are using your knowledge of cells to create a new technology that is going to address a problem that you are interested in addressing, okay? So to give you an example of biotechnology, there are some drugs that are used to treat patients and with many diseases that are formulated now with little particles, because they are so little, they are called nanoparticles, that are fatty, okay? And they are fatty because the cell membrane is made of fat. So what happens with that is that you facilitate whatever drug you are putting inside those little nanoparticles enter the cell because the fat that surrounds that particle fuse with the fat in the membrane of the cell. So you can deliver the drug. And that's a biotechnology in a way, okay? Bioengineering is basically using biology knowledge to engineer something important in the biological sense. So for example, I put here a heart, and one of the big problems that we have, for example, with a heart attack, with an infarction of the heart, is that the heart doesn't regenerate. It doesn't regenerate, so the tissue that suffer, the part of the heart that suffer the infarction, is going to form something like a scar, but it's not going to be functional anymore. So one of the things that they are doing in the field of bioengineering is trying to engineer cells that can, or structures that can be put there to facilitate the regeneration of the heart. So that is in the field of bioengineering. Also, for example, in some cases, if somebody is in the war and lost a leg in the war, for example, and you want to have a substitute leg for that person, you can actually engineer a leg that have functions similar to a normal leg. So things like that, and all of that is bioengineering, because you have to know knowledge, you have to have knowledge of how cells, the mechanical cells and how cells together move the body, okay? And finally, there is a field called bioremediation, which is really interesting, and it's becoming very important nowadays because we have a big interest in the environment. And there are a lot of things that can damage our environment. So for example, one of those things is oil. When there are oil spills in lakes, okay, what can happen? This oil is not going to be degraded very easily, and that damage the species and everything, the ecosystem in that lake, okay? So one of the things that scientists have discovered is that certain fungus can transform oil into things that are easily biodegradable, okay? Or can even use like as food by the organisms that live in a lake. So that is the field of bioremediation. So you can see there are a lot of applications of cell biology. In terms of jobs, well, you can work in academia, which is what I do, for example, in education, educating from any level, primary, secondary, higher education. You can be an educator. You can do research, as I do. You can work in science communication, which is the people that write textbooks, for example. And to write textbooks, like say you want to write a book about biology or about cell biology, you need to have knowledge to make sure that what you are saying is correct, right? But some people love doing communication and doing drawings and using art to explain concepts. So those are going to be working in communication. You can work in industry, so pharmaceutical industry, to study how new drugs work and see if you can use them for treatment of any disease. You can create medical supplies. You can work on cosmetics. So the cosmetic industry have a lot to do with cell biology. So why do you think we have ceramide as one of the things that women put on their face before they go to bed? Because ceramide is something that is produced by your skin cells, okay? It's like an oily layer that we have on the skin that is going to prevent the evaporation of water. And that keeps, they say, oh, keeps a moisturizer with ceramide. It's not that they are adding water to you. They are preserving your own water on the skin by that ceramide, okay? So that is cosmetic industry. And if you like cosmetic and interested in that, well, there you go, that's your field. You can also work on legislation. For example, the people that revise, there are lawyers that revise new patents for new products, maybe a product that you wanna use for patients, for example, or maybe a new technology you wanna use to do regeneration of the heart. And you need to revise that that is correct and that that is not gonna cause any damage to the patient, to the person that is receiving it. So you need a person that knows science to review that patent, apart from looking at the legal side. So there are scientists that became lawyers to do that, okay? And finally, you can work in the government as well. For example, making sure that science is working properly, that science is addressing the society needs, and all of that. If you are interested in that aspect and in leadership, then that's also a possibility. So you can see it's a world of possibilities. Now I would like to talk a little bit about my research, because probably, and I hope that you are interested in hearing a little bit of this. So I study the biology of normal and cancerous mammary cells, so cells that form the mammary gland, the breast in people. And I study how cancer cells that are that have formed a tumor, move from the original site where they are and to a different site, and what makes them form a colony in that site that made them grow and resist therapy treatment and then kill a patient. So those are the things that I am interested in. And I'm interested in biomarkers. Biomarkers are molecules that can tell you something about disease. So could be something that patients have in the blood that I can measure to say, oh yeah, this individual is prone to develop metastasis. So let's treat him with these drugs that are more potent to make sure that that doesn't happen, for example. So those are biomarkers. And I am so interested in molecular therapy because my research discovered molecules that are involved in metastasis. And then we are trying to target those molecules to prevent metastasis or treat metastasis, okay? So cell biology is the discipline that studies the structure, the function, and the behavior of cells. And you probably know that, and you probably learned that since primary school. But cancer biology studies the process by which a normal cell becomes cancerous, and then applies this knowledge to medicine, okay? So here you can see, so remember what I was mentioning, that cells organize to form tissues. So these are cells, we call them epithelial cells because they organize in layers, in very neat layers, could be one layer or could be several layers. When cancer forms, these cells lose that organization and they are not neatly organized anymore, they are not attached to each other, and then they become very disorganized and form a tumor mass. So that is the process of cancer. So we study what happened inside the cells for this to happen. A curious fact, just in case you don't know where the word cancer comes from, the word cancer actually comes from a crab, because this individual that lived before the Christian era, that is considered the father of medicine, Hippocrates, when he was looking at tumors, the way the vessels organized around the tumor reminded him of the way the claws of a crab organized, the way they look. And so he called these tumors carcinos, which is the Greek for crab, okay? And then it was translated from them to carcinos, which is the Latin word. And finally, it became the word cancer that we use every day. So that's where it comes from. So whenever you think you see a crab, now you know why the cancer of the zodiac is a crab and why the cancer that is a disease is also a crab. And so my research addressed several sustainable development goals. Remember that I talked to you about the United Nations Sustainable Development Goals. So my research is interested in improving health and well-being, in improving the quality of education, because with the knowledge that we accumulate, we can educate people better about the way cell behaves. And also, I am very interested in partnerships to achieve my goals. I do that because for us to advance our research, I collaborate with many people so that the research can be advanced a lot faster and we can have an impact on society a lot faster. So here is a picture of the people in my lab. So this is the Ontario Veterinary College. I work in a college of veterinary medicine. So the Faculty of Veterinary Medicine is here. And in the third floor, in this area here, is where my lab is located. Okay, my lab is my laboratory. And the girls that are going to be talking to you today doing the activity in the lab are there right now. They are going to be doing it there. And you can see how this area is inside. And those girls are Val and Cassidy. They are here beside me. You can see them. And these are other people that are in my lab. So all of these at the top are graduate students. And Sarai, who is actually Venezuelan, she is going to be a research summer student. And we have another girl who is Mexican coming in to join the lab in the summer. And so we have then this group of people working together to address different problems in mammary cancer as well as bone cancer. And you see these animals here because we do what is called comparative studies. They are called comparative studies because we always try to compare the way cells behave in cats and dogs to the way cells behave in humans. We also compare the way cells behave in cows because they also have mammary glands. We drink milk from the cows because of these mammary glands. And also why? Because when we compare, when we do our studies with all these different cells, we find common mechanisms, and they can be applied for veterinary medicine as well, because dogs also get cancer, cats also get cancer, okay? So we want to cure our pets as well. And that's why I studied these comparing cells from different species. And in the case of the cow, well, we are interested in milk industry. Milk industry is a big thing everywhere, right? People drink milk. So if you understand the mammary gland better, you can actually apply it to improve the yield of milk from cows, for example. So all of this is very, very interesting and very important for society. And I wanted to stop my lab presentation by showing you two things that you are going to find interesting, or a statue that symbolizes University of Wealth is the griffon. Okay, and it's a beautiful, beautiful mythologic animal. Everybody that comes to Wild want to have a picture taken with the griffon. And you probably noticed this logo here. This is the logo of my lab. AVP initially stands for Alicia Viloria Petit, the name of my lab. So everybody say, oh, they work, Cassidy and Bob, they work in the AVP lab. But there were, sometime in my lab, there were a group of students that they thought that AVP was very boring. So they needed to do something with that name that make it more funny, more interesting, right? So they decided that they were gonna be calling this group Aliens versus Predators, okay? And so my lab is now the Alien versus Predator labs. And I guess Bob and Cassidy will be able to tell you whether they are in the alien side or in the predator side. So it is something that I found very funny, and so we stick to it. And one of my students made the logo, okay? And this logo have become like really, really famous. Okay, so let's talk a little bit about the research now in the sense of what we do. You remember there is the name microscope somewhere in the title of my talk. And so I'm gonna talk to you today about two microscope types. I'm gonna talk to you about the bright field microscope, and I'm going to talk to you about the confocal microscope. So the bright field microscope is the one that you have learned when you study cell biology in a school, if you have, and maybe some of you even have the opportunity to see one in a lab that they have at the school. For example, I know not all schools have a microscope, but it's something that many schools have. So the bright microscope works with the regular white light, and then this light is produced by a lamp, and that goes through the specimen that you wanna see. So this microscope have mechanical and optical components. So the mechanical components are the ones that move the different parts of the microscope. For example, my eyes are very close together. My head is very small. So when I am looking at the microscope, I need to move the oculars toward the center every time, because any other person in the lab have eyes that are wider than mine. So that's a mechanical component. You can adjust the eyepieces close to you, so they actually align with your eyes, okay? So then you have the optical components. What are the optical components? It's basically a system of lenses, okay? So, well, you have the light shining, and then you have a condenser that, as the name indicates, condenses the light, so it's more centered on the specimen, okay? And then it passes through your sample, which could be, cells that you have, live cells, could be like cells that you have in a dish, or it could be a piece of tissue that you have, treated to be able to wash that piece of tissue that is very, very thin, and you can wash the cells in there. And so after it passes here, it goes to a first lens that is called the objective. And that objective is the one that gives the same amplification of the image, okay, and determines the resolution. The resolution is how needed you are going to be seeing that sample. And that is determined by the capacity that this objective has to see two very small structures that are individual structures, okay? So the resolution of this type of microscope is a maximum of 1,500 times magnification, okay? So you can see things 1,500 times larger than they originally are, okay? And it's determined, the resolution that you get is determined by the quality of the objective. So this objective then projects the image toward the viewer, but before you see it, it passes through the ocular. And the ocular is another lens that magnifies. It magnifies only, but it doesn't affect resolution. So really what determines how well it's going to look is this. This determines how big it's going to look, but this determines how well it's going to look, okay? So, so this is the microscope for you, and here is Val with Sarah E, and they are looking at cells, and in this particular case, the microscope is connected to a camera, and so this camera is connected to a computer, and two people can see the sample, one looking at the sample through the microscope, and the other one looking at them on the computer. And you can also take pictures and save your pictures and use them for presentation. In the lab, we also have another variety of this bright microscope, which is a digital one. And in the digital one, what happens is you don't see the sample through an ocular, but you see the sample through a monitor, because it doesn't go through the ocular, but after the image is amplified, it goes through a computer. Okay, so that's what happened, and you see it like here. And what I am showing you here is fat cells of the body. Okay, fat cells of the body, adipocytes, they are called. Okay, adipocytes. And what you see here in red are the big fat bubbles that they accumulate inside them. And you probably are asking yourself, why are we interested in looking at fat cells? Well, because it is known that women that are obese develop breast cancers that are more aggressive, okay? Especially if they are premenopausal, before they get menopause, okay? And they get they are more aggressive. Why they are more aggressive? Because they metastasize more. They move more frequently and to more organs than in other women. So we are interested in knowing how these fat cells influence the breast cancer cells to make them more aggressive, if it is that they produce something. And we are looking at those molecules that produce to see if we can identify molecules that are responsible for this, so that we can somehow eliminate or target those molecules. Okay, so in this particular case, what you are seeing is we took some cells that are pre-adipocytes and we give them nutrients, we give them things that we know are going to make them become adipocytes, okay? And so that's why over time they become adipocytes and their center is filled by a big back wall, a big bubble of fat. That's what happens. And so here you can see there before we stimulate them, and here you can see them when we stimulate them, how they become fatty cells. And then after we do that, then we culture those cells together with the breast cancer cells and we see what happened, okay? And we have, Cassidy have discovered some very interesting things that she's gonna be presenting in her. This is the fence, which is happening this April. So the other microscope I work with is the confocal microscope. And the confocal microscope is very interesting. It is a variation of to look at cells that have fluorescence labeling. So it's very difficult, as you can imagine, to look at molecules inside the cells and to figure out where they are, because where they are give us an idea of what the function is, okay? So for example, if it's a molecule that is in the nucleus of the cell, you can imagine that it has something to do with the regulation of the DNA of the cell, of how the cell, let's say, replicates, or how the cell is going to be producing proteins starting from the transcription and followed by the translation of a given gene. So that's why we are interested in looking at molecules in the cells, okay? And to do that, we use a number of tools. For example, we use antibodies that recognize specific molecules, and those antibodies are labeled with things that we call fluorochromes. And what these fluorochromes are molecules that when you shine light to them of a particular wavelength with a laser, okay, which is what a confocal uses, then they emit light with a higher wavelength. And that gives a fluorescent color, okay? And so we are able to capture that color with this technology. And then we can even use different fluorochromes with different wavelengths at the same time to be able to identify where different molecules, for example, the molecules here in red and the molecules in green, where they are in the cell. Are they working together or are they separated? And then the red one here is obviously in the cytoplasm, and the blue here indicates the nucleus, because we stain the nucleus with a molecule that is fluorescent by itself and it binds to DNA, okay? So that's really, really nice. And that's why you're seeing the nucleus here in blue. And how is this working? Well, this works in a very, very sophisticated way. So you have a laser, which is a very potent light that is emitted in a very narrow space. So there is not much light wasted into space, okay? And this laser is shown through a very small hole that goes through a system of mirrors and then goes to the sample. It goes to the sample and then goes back and goes through a very tiny hole and then goes to a detector that shows in the computer, okay? And this system allows for us to use a very focal, that means in a very, very tiny area and in just one plane of our sample, very focal direction of the light so that we see things really, really precisely, okay? And so by doing this, we can see things so neated, as you see here. And by combining this with these fluorochromes and different wavelengths of lasers, so our confocal microscope have 4 different lasers, so we can try the 4 different colors.
Señora.
And then you can see this,
Bueno.
okay?
Oye, pues.
Can we, somebody have the microphone on? Can we turn off that microphone, please? Okay, so this is the way we can see the molecules inside the cell. So remember, this is important how this confocal microscope work. And here is a confocal microscope. We have a similar one in our lab, not in our lab, but in the floor that is used by several researchers in our floor. And I just wanted to mention, using this image, you have all the elements of STEM. Because you have science, you have cell biology, you have immunology because we use antibodies. And antibodies, we know about antibodies because of immunology, right? And we use photonics, which is a part of a branch of physics, okay? Because without photonics, you cannot make that machine work if you don't have a knowledge of how light works, okay? And then we have technology, because confocal microscopy, whoever develops it, is a very, very powerful technology. We have engineering because we have an electronic and mechanic systems. We have a software all together. So there is electronic engineering, there is materials engineering, there is software engineering, because to be able to analyze this, we need a specialized software. And the mathematics that is involved in creating all of this, the software, for example, use a lot of geometry to localize things in space, for example. So this is a powerful technique. So my contribution to of my work to society is important, and I already mentioned it. So I am interested in contribute better tools to diagnosis, prognosis, and treatment. So that address the number 3 of the Sustainable Development Goals. I also try to use products that are not so harmful for the environment. I try to order as little as possible so we don't waste. So in that case, I try to have responsible consumption and production. And because I educate, and I have learned a lot to educate, and I continue to learn to educate, then I would like to think that I am promoting to quality education. And because I promote women in science, then I would like to say that I am promoting gender equality and reducing inequalities. So this part is more about, you know, what obstacles I encounter in my career and what opportunities I encountered, that both of which were important for me to grow and to be where I am now. Because you probably are asking yourself, okay, is it easy to go in this path? Because you see, it's fun. I have a lot of fun working. I love this stuff. But then there are things that were difficult. I cannot lie about this. For example, finances were difficult because, you know, do graduate school is expensive. And the language was difficult. I mean, I have to learn English when I was 26 years old. I haven't spoken English before, and I still make mistakes, but I feel able to communicate, and I can communicate well. But
Hello, everybody. Welcome to this science lesson. We're going to be having interesting information. I'm Laura, and I will be hosting this session. So, hoping everybody is connected already. I'm going to pass it to Oriana. Welcome, Oriana.
Hi, Vicky. Welcome to our last science workshop session. Today, as Laura said, we are going to have Linda Jacobo, who is going to teach us about eco-toxicology. And well, now let's get started with the rules. First, check camera connections. Check connections, camera, microphone, router, or any hardware to be used for video conferencing. 2, have a good lighting. Try to have good lighting either with natural or artificial lights. Third, identify yourself. Remember to rename yourself on the Zoom platform, indicating name and surname, country of residence. For example, Maria Torres, Mexico. 4, choosing the right an appropriate background. Choose a simple background, avoiding any bright, exotic, or messy, dirty background. 5, choosing an appropriate wardrobe. Dressing correctly and completely is important because any movement out of the frame can reveal something that should not have been seen. Have a good framing. We must look for a certain framing with a certain margin of movement so that we can be seen correctly. 7, avoidance of ambient noise. It's very annoying to hear noises of all kinds when someone is talking, bricks, banging, music, discussion, television, or radio in the background. 4, start on time. Be on time so that the meeting lasts the scheduled time. And finally, taking turns and following the agenda. Follow the order of the day and avoid deviating from the agenda. Respect turns to speak and questions, avoiding interruptions.
Great, Doriana. Thank you for reading all the instructions. I hope everybody understood. You are already familiar with them. So now I'm gonna introduce our speaker, which is Linda. Linda is Mexican and currently lives in the United States. She is an assistant professor at the School of Public Health of San Diego State University. Linda has a PhD in toxicogenomics from the Institut National de la Recherche Scientifique in Quebec, Canada. She has more than 10 years of research and teaching mentoring experience, including work with Indigenous and rural communities. Her work aims to connect health, environment, gender equality, social inclusion, and diversity towards a sustainable future. She's also a member of the HV Women in STEM Leadership Program, acting against climate change on a global scale and observing its effect in Antarctica and increasing women's leadership. In 2021, Linda received the Hispanic Women of the Year Merit Award in Canada. Linda has been an active participant in Geeky Latinas since the beginning of the program, always transmitting her enthusiasm for everything she does to our team and students. As always, we feel very happy and honored to have her with us today. So welcome, Linda, you have the floor.
Thank you, Laura. And hi, everyone. I'm just gonna start sharing my presentation. Let me know if it works. There. Everybody can see my presentation.
Great.
Yeah. Okay, perfect. Just gonna move this over here. There. Okay. Well, good morning, everyone. I'm just waking up because for me it's 7 a.m. So it's really early. So I'm happy to be here. I'm really excited. Today we're gonna talk a little bit about what is ecotoxicogenomics. That's one of my fields, and it's what it's about it, what we do as a scientist. Right now, I'm a professor in STSU and public health, so I live in California, and I'm going to share a little bit of my favorite spots in California. So you can follow me on social media. I'm always publishing things related with the STEM, women in STEM, and I'm going to be sharing my journal to the Antarctica. Just 1 second. There. So I'm going to take a little bit of my background. So I was born in Tijuana. I'm a person from the borders. So I'm between, like, I grew up like in Mexico with a little bit of the, well, a lot of the influence of the US. And this is the land of the Kumeyaay land. And so I'm from this region. I'm from this Kumeyaay beautiful land. And this is the indigents that they are here in the north of Mexico and that we share with the United States. It's a binational indigent culture. So this is the water, the first one, the first slide. I don't know if I can, I'm just gonna take a pointer, like just 1 second. Where can I take, maybe notes? No. Voila, I just found it. I guess everybody can see my mouse, right? Okay, so the first one is, this is the border. This is Tijuana, where I was born, and this says US. This is San Diego. So as you can see, like, we share like the border, but we also share a lot of the culture. So it's a really peculiar culture. We like to call ourselves as a transbordery like culture. So it's really peculiar. It's really different to other regions, but we're really proud to be part of this communication between those two entities that we share. So those are my parents. Both of them are scientists. So when I was a little girl, I always dreamed relate like everything related with science. My favorite animals were dinosaurs. So my first microscope, I have it when I was 4 years old. So I grew up in this family, in this environmental, surrounded by science. That was my life, pure science. You know, like making experiments with my mom. So I grew up in this kind of life that I really enjoy a lot. This is me when I was a little girl doing experiments. And so I will say, like, I never thought that because I was a woman, I would not be able to study science, because I have a big example that it was my mother. And later, when I was older, I noticed that I will have some challenges because of this, but I was not prepared to face those challenges, because in my reality, it was okay to be a woman scientist. So see, this is the beautiful Baja. This is Baja California. So I was born in Tijuana, that is Baja California, and we have San Diego, where I live now, and that is California. So you see, like, we even have a name, similar names, but this is Baja California. This is the Sea of Cortez, and Gulf of California, we also call it. And this is the Jacques Cousteau Aquarium. It's beautiful. You have whales, you have a lot of animals, a lot of fish. It's beautiful. It's one of my favorite places, spots in Baja. It goes Bahía de Los Ángeles. It's wonderful. So my professional achievements, well, I started my career as a scientist in UABC, Universidad Autónoma de California, in Tijuana. So I study chemistry, pharmaceutical biologist, and yeah, I started like having some contact with what was chemist, what was science, what is biology. I started doing like some tests with microorganisms. So I have to do like my internship. We have to do 2 internships. One, it was in a hospital, that we work in hospitals. My career is for working in hospitals. So I was working in a hospital, performing like tests, urine, blood, taking blood from patients. It was really stressful for me. And the second part of my internship, it was more like a research project. So I developed a project with organic chemists, and that was, I really enjoyed doing research. So that was one of my first research, and I was really excited to have this opportunity. Later, I started like a specialty in Simbesta Irapuato. So I just like this study a little bit of genetic engineering. I have this opportunity as a summer research project, and I really enjoy it. I was like kind of new, and I knew that I would like to study like genomics, but maybe like a little bit with something different, because genetic engineering is really complex. I mean, not because it was complex, it was like, it's really complex. There's a lot of controversy related to that. So I knew that I wanted to study genomics, but maybe not related to plants. I wanted to focus more in toxicology. So later on, I studied my master's degree in CCS in Ensenada, that is also in California. It's just next to the water, next to the Pacific Ocean. And I started studying with microorganisms, and I changed a little bit to shrimps, and I finished working with crabs and metals. So in that moment, there was nothing related with ecotoxicogenomics. It was just ecotoxicology, that it was already like a big combination of things, but nothing related with genomics. So after that, I worked, well, this is some pictures of my master's. So we used to have like the crabs separate them in this small like caves, and then they start like so I studied like how the metal contaminants in the water will affect the developing of the crabs. So for example, this is what normally happens, is molting. So when they molt, usually you have like a soft kind of exoskeleton, but it doesn't like it. Usually with time, it will change. It will make it like a thicker, stronger, resistant, you know, like I think everybody has touch in their lives, like a crab. It's really strong. But in this case, because there was metals in the water and the way absorbing them, they will never make the exoskeleton like stronger. It will just be soft. So that make them an easy prey for the predator. So yeah, that was a big issue. So we can see how our presence in the presence of the coast, you know, like there's boats, it's extractions of different minerals, it'll affect the ecosystem. So that was my research project. I also did a research project with shrimps, because we produce a lot of shrimps here in the Pacific Ocean. Well, this shrimp actually, it comes from the, okay, I don't know if you remember Baja, but we have two oceans, well, two waters. We have the Pacific Ocean and we have the Cortez Sea. So these ones are from hot water. So hot water is more the Cortez Ocean. The other one is colder. So shrimps are in the really hot weathers. They love warm. They cannot support like really low temperatures. So these ones, we have some issues with some viruses. So we modify a virus for ecological niches, and they can just like substitute the ones that are pathogens for the ones that are not pathogens. So it was really interesting working with the shrimps. We have to inject them. So it was really complicated with the shrimp. But yes, so there was a lot of challenges, but we did it to protect the ecosystems. I also work with this kind of snails, and these ones are the sea snails, and they are toxic. So we also produce, you can develop medication with these venoms. So yes, with this poison thing. So we produce we develop, first of all, you have to elucidate. It means like you have to understand what is the poison about, like what is the structure, the chemical structure, what are the potential pharmaceutical products that you can create with that. So we use it for creating medication related to painkillers. Like we try to make painkillers, but it's complicated. Yeah, this one is el botete. That's really famous here in the Pacific Ocean, and yes, and it's toxic only from the liver. So the liver is the one that is really toxic. So we need to be careful about. So we need to study. In that moment, I was studying it like why the liver is toxic, and it's not related to the spines. It's not related with the thorns. Like what is the toxic about that? And the last one is I was taking blood from my crabs because I was doing research related with the blood to see if I can quantify the metals in the blood. As you can see, the blood is clear, and when oxidized, it's kind of blue, because they don't have hemoglobin like us. They have hemocyanin. It means that they have copper instead of iron in the blood. And I also have another experience. Let's see if you can see this. That is a shark. This is a shark from the Pacific Ocean. This is a Californian shark. And we work with these animals to produce some antivenoms. As you must know, California and Baja California, we have several issues with poison animals. They kill a lot of people every year. Mostly like people who is, for example, farm workers, so they are on the field all day long. So yes, we produce antivenoms from rattlesnakes. We have a lot of rattlesnakes here. We have a lot of spiders. I will show a picture later of my work related to toxicology. But yes, that's really, yeah, that's a really big deal here. I would say it's the same in Arizona and Texas. They have the same issue. So yes, later I was assistant professor in UABC in the campus Tijuana. And yes, I was study like a lot about poison animals in the region. I focus mostly in my region because my region has a lot of animals. And yes, like in the south, they have more mushrooms, toxic mushrooms, spiders, scorpions, yeah, like rattlesnakes, and a lot of plants that are toxic. So those are my students. And yes, I took them to several field work. So they were really excited to make some field work. We went to the beach, we take some samples. So this is the spider. I hope all of you recognize this one, right? It's the black widow. And this one is cosmopolitan. It means that it can be everywhere, but still, it is really dangerous. Here we have this plant that we call is the one that it makes the castor oil that we call resino. This is the resino oil, but they call it castor oil. I don't know. And yeah, we have a problem with some lakes. We have artificial lakes. Tijuana is like kind of Mediterranean kind of weather. It's really peculiar because you see the desert, and after the desert you see the beach. It's you see the big ocean. As I show in the previous picture, you know, like the beautiful landscapes. It is that. It's like you have desert and then beautiful oceans, beautiful landscapes with mountains and beach. And we produce a lot of wine, right? We have a really similar weather to the south of France, and yeah, between, you know, like that's why they call it. Actually, the immigration that we have in the north of Mexico, it was mainly people who produce wine, and that's the reason why we conserve it. And yeah, and they call it Mediterranean, even if this is not the Mediterranean region, but we call it like that because it's really related to the other one. So this is a plant that it calls, maybe you know this plant is burundanga. That's a really famous plant. We have a lot of here. And yeah, actually the toxic chemical is toloache. And the indigenous women from this region used it to control men. So they used to make a tea, and then in this tea, they give it to the men. And yes, and it will lose their willing. So you can use it for control the men. It was an indigenous tradition from this area. What else? This is the scorpions. You can use a lamp, UV lamp, if you want to see them. This one is a toxic, a venom one. You can recognize it because, you know, the pedipalps that they have here, they are really thin, but the tail is really big. So you can recognize them easily, and they're really tiny. They're not big, and they're clear. You see them in the morning, you don't see anything. They're really clear. So that's why the reason why we have to use a UV lamp. It's really easy to see them. Actually, every time that I go to field work, I try to have a UV lamp and just double check first if there's any scorpion, because as I mentioned, in this region, we have a lot. So we always need to be careful about it. I also check my shoes every time that I wear them, because there can be spiders, there can be scorpions. It's kind of a little bit like Australia. You know, you have to be careful all the time. Yeah, this is not a poison snake. This is just a baby fake rattlesnake, but it moves like it moves like like a rattlesnake. It moves like the tail, like making like using the rattle that isn't existent. But yeah, yeah. So, and this is all the animals that we collect with my students. Yes, several. A lot. A centipede, scorpions, different kind of. And this one, I don't know how you I don't know how you recognize, like how you say it in your region. Probably, I think this one is everywhere, but here in the north, we call it Niño de la Tierra. I don't know. They call it Niño. It's just like that. Apparently, it makes a noise, and it seems like at the face, it looks like a baby. I don't know. It's kind of creepy, honestly. But yes, it's Niño de la Tierra. We call it like that. And there you go. That's the one of the. It's not toxic. It doesn't produce a toxin, but if you touch it, it's like kind of urticant. So that's the like an astringent. You know, like it's kind of like that. But yeah, it's not toxic per se. Yes. I used to say rattlesnake a lot. I love rattlesnakes, and there is a big issue here in the region. Like a lot of people is afraid. Of course, they're afraid. They poison, but voila, like the students used to save some rattlesnakes and just bring it to my office. And in this case, we try to feed because it was a baby. You can see it only have 1, 2, 2.5 rattles. So yes, it was a really young and really small one. So at the beginning, I tried to give them pinkies. Pinkies are the baby mice. But he was like, I didn't check it was a he or she, but it didn't like appreciate this pinky. So what I started doing, it was just like, okay, we're gonna make a test with a mouse, and let's see if it works. And it works. So I guess like they prefer to eat like moving animals, just to be sure. Remember that if an animal is already dead, there's some animals that they are eating dead animals, right? We have some of them that they eat, but no snakes. Snakes need to be sure, like if they are alive, because if they're dead, it means that they have a disease or they're sick. So they are really precautious about what they're gonna eat. So this one, we feed them and then we release it in the ecosystem because they need to be in their own ecosystem. This one, it was another rattlesnake. It was a big one, a massive. It was one of the biggest I have seen in my life. It just appeared when I was walking. I was doing hiking, and there you go. You have a snake there. It's really common, honestly. You have to see the size of my boots. They're really big because you have no idea when you can see a snake, a rattlesnake. Yes, here we did some field work in indigenous community from this region. So in Baja California, we share the indigenous community with California. We call us Humanos. That's the group of the ethnicity is Humanos, and we have 5 different indigens. So one of those communities, I used to bring my students to this community so we can perform like health services to them. So those are my students, and they were doing blood tests and helping the community. Unfortunately, this community has severity of pollution with pesticides. So we were doing some genotoxicity tests also. And also, like, as you must know, some communities in the region, indigenous communities, they use the water from, like, they use kind of like this, kind of like, I just forgot the word. But they use water that is not like clean, right? Like it's not like a potable water. You have to buy it by yourself. Wells, or you use it from wells. I just remember it. So you use wells. So this is what they're doing. So we were testing the blood, the blood of the women, because the women have they lose their teeth really young. So that is a symptom of pollution with metals. So we were testing the water for metals, and see there's a correlation between the amount of metals in their women's blood versus the water. So yeah. Okay, so finally the snake finished eating the mouse. Okay, so after that, like I traveled around the Mexico, so to different spots, studying different toxic things. I started here in Mazatlan, Sinaloa, where I studied like phytoplankton that is like kelps, by these mini kelps that they produce the red bloom algae, like algae bloom. So the toxic ones. So yes, I was studying there, like what we can do, how we can detect it, because usually when there's a red bloom, you have to like make a big alert in Mexico, so nobody like nobody there. There's no fishermen. There's no fishermen in the water, and you don't fish any animals in a moment. Later, I went to Puebla to work with the bats. There's bats in caves, and I also traveled to Chiapas and Guatemala to be in the Selva Lacandona, so the Lacandona jungle. I did some field work there with different scientists, so we can learn techniques for field work. And this is some pictures about those expeditions that I did. So this is the toxic, this is a toxic phytoplankton. Believe it or not, these ones are more toxic than the rattlesnakes. So it's really dangerous. Yeah, tarantulas. I did some work related with the monkey. This it calls spider monkey. This is the spider monkey. Yes, it's kind of they're kind of aggressive. We were trying to camp close to them because we need to collect poop to see what they eat. So you collect poop because it's a non-invasive way to study the spider monkey. So they start throwing us poop, right? It's like a mechanism of defense from the spider monkey. This is the bats. This is like a photo trap. Usually like it detects movement, and when there's movement, you can take pictures automatically. You don't need to be there. So usually you put like a trap in like you put like I don't know, like a bail, you know, like usually for I was working with jaguar, and the Mexican jaguar that is really powerful and beautiful, and it has a big representation for the indigents, the Mayas mainly, and we were trying to see them with a photo trap. So we need to put like a veil, and the veil is made with tuna. So it's really smelly. You know that tuna is smelly. It's like everywhere. You can detect it like miles around. So you just like put it there, hang it from there, and then just like connect the photo trap. And every time that like it moves, it will take a picture. So you can just let it just go, take the memory, and just see what you catch. Well, not you catch, but what you catch in the camera, right? This is not invasive, so you don't touch the animals. We usually try not to touch the animals because we don't want to disturb. Sometimes it's necessary to take samples, but the majority of the time we don't take samples. We just take pictures or poop or urine samples, nothing that is invasive. This is toxic mushrooms. Yeah, this is the poop. This is a toad. This is a toxic one. This is the poison glands, and yeah, and this is like they found like a dead spider monkey dead. So just like take I guess like the skeleton and just produce that. Let's see here. So later I started a PhD between SDSU, that's the university where I work right now, and Tijuana, related to persistent contaminants in the wastewater, and also related with wine. But I didn't finish it. After 1 year, I just quit, and I decided to move to Canada. So I earned a grant where they pay me everything to study. So I start my PhD in Queen's University in Kingston, Ontario. Yes, it was really excited. I started there for 1 year, and after 1 year, I just moved into Quebec. to the other institute, the Institut National de la Recherche Scientifique, and that's a completely different province of Canada. They speak French, and yeah, it was a big challenge because my spec was none. So yeah, I have to learn French at the same time that I was like doing my PhD, and I will tell a little bit about that later. After that, I moved back to Ontario to do my postdoctoral researcher. This one was in the University of Ontario Institute of Technology. And at the same time, I have some field works in the Institute for Sustainability Development in Winnipeg, Manitoba. That this is a unique area for experimental lakes. It's a unique in the world, so it's really famous. And I have this opportunity to be on field work there, and wonderful landscapes, wonderful place. But there's really important the work that they are doing there, because as it's unique, they have the opportunity to just perform realistic oil spills or scenarios related to pollution in the water. After that, I moved back. After my postdoc, I moved back to Quebec, and I was working in a company that it calls Bureau Veritas. And yes, and I was working there as a toxicologist, as a scientist. So I work in the certification, for example, like the companies, if they can release water to the lakes or not related to the pollution. So my job was to be sure that they are not polluting, companies are not polluting the ecosystem. So it was a really hard job. It was really about the developing of policies, laws, regulations. But yeah, it was really interesting. I really enjoyed it. And after that, I just got my job in San Diego State University. So I go back to California after having -40 Celsius. I just go to +40 Celsius. So it was a big change for me of temperature, of culture, you know, because this one, it was more French culture. My partner, I met my partner there. So my partner, he's French. So it was like, yeah, I was more used to like to speak more French than English. I used to have both because, you know, talking with Canada, when you talk with the federal government, you have to talk in English or both languages. But in Quebec, you have to only speak French. So I was like kind of all the time speaking, they call it Frank English, you know, like you mix French and English. But in California, no. In California, nobody speaks French. I also speak Spanish, obviously. So like I was able to now to speak my own language, Spanish, and mix it with English. So now we speak Spanglish. I came from, so I've been changing a lot. So now in California, there's a lot of people, you don't even have to speak English anymore. Like only in the school, of course, I have to speak English because it's really international. But I will say like a lot of the time I speak Spanish in California. But there's a lot of Mexicans mainly, like mostly they're Mexicans because we're so close to the border. But yes, so I'm so happy to be able to speak Spanish all the time. So this is my university. This is the Hefner High, and this is the Department of Public Health. Actually, this is one part of the campus. This is the library. Yeah, we have we recently we have like a celebration for our 125 years of this school. So it's a really old school. Those are my favorite spots in San Diego. This is a beautiful garden in Balboa Park. And this is La Jolla Cove. As you see in California, everything is Spanish. You have La Jolla, Chula Vista, San Diego, San Isidro. This one is Balboa Park, and the style is really like a Spanish, you know, like usually in the north of Mexico, we don't have these structures because the Spanish didn't arrive here in the north. But for example, in the center of Mexico or Guanajuato, or those regions that are more centralized, they do have like really Spanish structures. So California took that architecture and they put it in San Diego. So they have like a lot of old buildings, builds kind of this Spanish colonialized architecture. And yes, but so it's kind of like that. It's beautiful. Honestly, they have beautiful museums here. Balboa Park is one of my favorite spots. They have museums, yeah, like theaters where you can go to see beautiful place. Yeah. And yeah, in La Jolla Cove, you can see these little guys, the lions, see all the time. All year long, we have sea lions. They're beautiful, they're wonderful, they make a lot of noise. And it's wonderful when you go running and you go hiking, you just can see these wonderful and majestuous animals. Okay, so I'm going to talk a little bit about ecotoxicogenomics. So what it means is like there's 3 words in one. So we have ecology first, we have toxicology second, and we have the last one that is genomics, right? So it's a combination between ecology, toxicology, and genomics. And what it means is like using tools related to genomics or toxicology purpose in an ecological aspect. So it means in the ecosystem. So what is the central dogma of molecular biology? I'm just going to mention this one. I know it's like maybe too much, but you know DNA, right? So we have DNA that it will transform to RNA, and then it will be translated to protein. So it's like DNA is one language, and in order to be read to produce the proteins, you need to translate it to another language. So DNA and RNA are different languages. It's like kind of Spanish and English. So we have a person who is translator, everything from DNA to RNA, and then the RNA can be understood by another component to produce a protein. So that's the reason why it's so important that there's no damages between those 3 aspects. Otherwise, the proteins are not going to be exposed, but they're going to be built because they are built in a right way. And in toxicology, we have 3 severity, like toxic consequences, or like, yeah, consequences for toxic poison or toxic products. And we have mutagenesis, that it means that in DNA there is like change. You affect the DNA, right? So for example, like you have the normal one, and then there's like a change of something, you add something, or you just eliminate something. So it can be transforming the DNA. So it's really important that our DNA is not transformed, but it can be transformed by several things, by the UV, like it can be transformed by several things. So mutagenesis is a change in the DNA, okay? And then we have carcinogenesis, that it means that it can produce cancer, right? Like the creation of cancer. And carcinogenesis is, well, like formational, like, yes, producing tumors, right? Producing tumors, not all the time it means that it's a cancer. It can be just a proliferation of cells, but it doesn't mean that it's cancer. To be cancer, they need to be mutated. You're right? Like you need to be mutated to have cancer, like to have a carcinogenesis. So voila, like we have this one. They can be 2 kind of cancers, the ones that are produced by mutagenesis or the ones that are affected in the cell cycle. We also have teratogenesis, that is produced by a mutagenesis. So we have a mutagenesis that it can produce teratogenesis. Teratogenesis came from teratos in Greek, that it means monster. So it's like when there's like a pregnant woman and there is an exposition during the pregnancy. This one is only during the pregnancy. During the pregnancy, the woman is exposed, like the developing of the embryo is exposed to contaminants. There's going to be a mutation in the DNA that is going to be irreversible. Teratogenesis is irreversible. There's no coming back from that. And then you have like this one, for example, we have production of bicephaly, that you have 2 faces, like 2 heads, right? Like in this case, 2 heads. And this one, for example, it happened after in the Gulf of Mexico. You know, I'm talking like we have several oceans, like in waters in Mexico. We have the Pacific Ocean, we have the Californian Gulf, and in the other side we have the Mexican Gulf. There was like around of like more than 10 years, 20, I'm thinking, there was like an oil spill. So they used like a little chemical compounds to try to eliminate the oil from the ocean, from the Gulf. And after that, it appeared this kind of animals because they were super toxic. So they were producing teratogenesis in the animal species. So we also have here bicephalis in totals. So yes, mutagenesis can produce teratogenesis, but it's only during the affections in the embryo. So we call this embryotoxicity. Okay. So we can have different kind of mutagenesis. As I mentioned, UV is one of those. So we can have chemicals that it can be drugs, for example, medications. Thalidomide was a drug that it was used in the 60s for pregnant women. So pregnant women, we take this one because apparently avoid that you feel dizzy, you feel the nauseous, you feel all the symptoms related to the pregnancy. But later, when the baby were born, you have mutagenesis, that in this case, it was teratogenesis. So there was a big study into it was a German company because of this. So after this medication, we learned that some medications, some drugs, they can cause and produce teratogenesis, and there were regulations about that. So now you can take any medication and you will see a pregnant woman and you will see like kind of a prohibited, right, ban, not use it for pregnant women. But it was only after this. So always like that, we make regulations after a big disaster. We all have also chemical compounds like acridine. This is like the drugs, the bombs that they use for war, that they are produced like, it's kind of a pesticide what they use. So yes, and oil can be, different kind of things. Chemicals is like any chemical product that it can be mutagenesis. Physical is like UV light, right? It's like a physical thing, like it's like radiation is physical. We also have a X-ray, like you don't touch it, right? We don't touch UV like rays, you just feel it. So it's kind of like that. Physical sometimes is the most dangerous because you don't feel it or you cannot touch it. It's not touchable, but it's really dangerous. It is really dangerous. And for the last, we have biological, that is microorganisms that they can produce cancer. So in this cancer, we have Helicobacter pylori, HPV, the papilloma virus, human papilloma viruses, and those one also produce cancer. So it's like an aspect that they can, like a microorganism that can modify your gene. It's really dangerous. Like it's wow. This one are really recently, because before we didn't know, but it's a really shocking thing. So yes, as I mentioned, mutagenesis, and we remember this guy Hulk, right? That he was actually affected by gamma radiation. I don't know if you remember the film. If you don't, you can go and see the film. But it's like that. It was a gamma radiation, poof, and just transforming a Hulk. It's kind of it's just a comic, but it's like really interesting how we know that there's radiation who can produce mutations in our DNA. And actually, I think in the film they say, right, like the gene was changed and things like that. And we have a big accident, Chernobyl accident, probably all of you know, in 1986. Yes, so there was like the Chernobyl nuclear plant has like apparently like explosion, and it just released radiation that until now, after 37 years, nobody can still live in that area because it still have a lot of radiation. So they have a lot of people with cancers, malformations in this case, I think it's a pig. So in the animals, in the farm animals, there was a lot of like malformation. So you can see how the big impact of the radiation in the ecosystem, right? Like nobody can live now there because it's still radioactive. We have another one that is a chemical. In this case, as I say, you need to touch it, right? It's a chemical product. So I just put like Spider-Man because it just reminded me, like, apparently it was a poison spider who just was injecting the venom inside this human, and you just have a Spider-Man, right? Because it changed the DNA and changed everything. So it's like that. You need like to touch it. You need to get approach to the chemical compound so it can cause a mutagenesis. So here we have a phenanthrene, and that is in different products. It can be in the meat, it can be in the cookies, it can be in the butter, and yeah, And there's been several studies related to that with mice and flies. Oil, of course, oil is one of those. Oil can also produce mutagenesis, and I'm going to show you some of my experiments during my PhD. This is a frog. This is a larvae of a frog. So this is the control one, and this is the one exposed to oil. So you can see that there's a big malformation in the embryo, right? Like this is like a normal one, the control, and you have here completely deformed the bird, the gut is not developed, you have several deformities. This is another kind of malformations. We have the cardiac tube malformed. There's no connections between the heart and the cardiac tube. You have a pericardial edema, these ones. You have craniofacial deformities, and you have like vertebral like malformation. This is the heart rate of a larvae. You can see the heart. So usually with those ones, the heart is really different. Like usually you cannot see the heart because it's so quickly that you just see like a pump that is distributing the blood. In this case, is you can see the whole time because there is like a cardiac tube that is broke. Yeah, this is the embryos. This is when they were fertilized. So when they're fertilized in stage 0, it's like that. Yeah, then they're developing. This one is curvy. Yeah, so you can see like different kind of malformations. Then we have mutagenesis, like are produced by oncogenic bacteria and oncoviruses. So I mentioned that Helicobacter pylori is one of those. Hepatitis virus is also producing cancer in the liver. So yes, it's like bacteria that can produce cancer or virus that can modify the DNA to produce cancer. So I know that I mentioned all these mutations are bad, but all mutations are bad. What do you think? No? I will let you think about it a little bit.
I think Elba Brito from Venezuela
Okay.
wants to answer.
Yes, go ahead.
Well, I think that it is not completely bad, but not completely good, you know? It can be good to advance sciences and that kind of things, but at the same time, it is not good to the thing that is having the mutation, you know? It will depend, I think.
Okay, good one. Very good. Exactly. It will depend for sure. No mutations are bad. Like we depend on mutations for evolution, right? Without these mutations, we cannot evolve to someone else, to something else. We did it from the monkey, right? Like from gorillas. And in the case, for example, of the birds, is the same thing. They evolved because of the need. There was a mutation in the DNA, and then they continue inherit this mutation. So this is the, for example, we have here the finch that is the ancestor one, and then they develop another a different eating patterns because of the evolution. So they mutate. And this one, for example, they eat insects. This one is like carpenter, so they need a really strong beak. And then we have the seed ones, right? The ones that eat the seeds. So they need like a curvy kind of beak. So as you know, evolution depends on mutations. And thanks to that, we have biodiversity, because we have the biodiversity in the peaks of our own birds, right? So it's really important that we continue having mutations. Not all mutations are bad. Obviously, if the mutation is because of contamination, it's not good. That's not good at all. But if the mutation is because of natural selection and you need it for evolve, go ahead. Like we need those. And we are evolving also. We are changing our DNA depending of the natural where we live, depending on our environment. So, okay, no mutations are bad. So right now what I'm doing, what I teach public health. So I focus more in different things, more like about poison animals, epidemiology of poison animals, because epidemiology and public health is kind of one of my priorities right now. So one of my research that I performed was water-based epidemiology. So it's like, for example, if a person is sick, even if they have symptoms or not, they're gonna go and shed in feces the virus, and then they're gonna just, the feces are gonna be transported into the wastewater treatment plant, and this wastewater influents are taken by their own plant or by ourselves, and then the analysis of wastewater samples, we analyze them for wastewater samples for SARS-CoV-2 or different diseases. So that way we can predict the amount of people who, even if they if they have symptoms or not, nevertheless they have symptoms or not, they gonna eliminate, they're gonna shed the virus. So we can predict that. So that's what we do right now. So we tracking diseases all the time. That's what my job is, track diseases. So I can prevent outbreaks. So yes, so this is what I do. I do RNA extraction. So I work with RNA, with DNA all the time, and proteins all the times. That's my expertise. And in that I can detect RNA, and I can just community monitoring, prevalence estimation, and I just can make implementations of public health guidelines, right? I can give advices and supervise public health to see if they're doing a good job. We need to prevent another outbreak in another community. So that's my job. And of course, like teaching classes and doing research with my students. So this is one of my last works. This is related with the proteins in the wastewater. So we have SARS-CoV-2, and I was able to detect all these sequenciation of proteins. Well, this is peptides actually, but this is a protein, and a peptide is just one part of the protein of this. This is the SARS-CoV-2 of all the structure of the SARS-CoV-2 virus. This protein is related with the replication. So this one, it was a really important one, and yeah, I was able to detect it. I also work a little bit with microorganisms, detecting proteins for the microorganisms, and also related to diseases. So this is Helicobacter pylori. I detect different kind of proteins related to cancer because of the presence of Helicobacter pylori. Also proteins related to the human papillomaviruses. The most dangerous are the 18 and the 16. Those are the ones who can produce cancer. And Oriana, you have raised a hand. Sorry.
Don't worry. It's because there's a question in the chat for about 3 presentations before, 3 slides before, where she's asking one more there. She's asking, what does letters means?
Okay, those are the names of the proteins. So this is the membrane M. This is the ORF3A. That's the name of the protein. This is a spike. So this is the spike protein. This is the ORF8 nuclear protein, and the ORF7A. Those are the names of the proteins. Thank you. And let's see. Oops. Okay. So more recently, I've been doing the surveillance of different diseases. So, for example, H5N1. That I don't know if you have the same, but we've been facing this one. This is the virus, the avian flu. So it's causing a lot of problems here in the US. The monkeypox, tuberculosis, poliomyelitis, immunization, prions, illicit drugs, climate change, microorganisms, and virus biomarker associated to biomarkers. So yes, and those are the things that I'm doing right now, tracking in the wastewater, and not only in the wastewater, most recently I also doing in the rivers. So here in California, this is like another area. So this is San Diego County, and this is Imperial Valley County. So we have a river who goes from Mexico, Mexicali. So San Diego County is in the border with Tijuana. Okay, San Diego, Tijuana, and Imperial County is with Mexicali. So we share rivers. So for example, this river, the new river, it starts in Mexicali. So it goes all up and is distributed into the Salton Sea. Salton Sea is a really controversial situation right now. We tried to recover it. It hasn't been possible. It's been years. And the problem is because of the contamination, all the pollution that arrived for years, now it's impossible to recover it. Not only that, every time we have dryness, you know, there's drought in this dryness in the area, there's drought in the region. We don't have water. California doesn't have water. California, Baja California, we don't have water. So every year, like they just discuss, it's like a spots of dryness in the sediments. And these things, because we have a lot of air, what it's gonna produce is like with the air, just like distribute those contaminants that they were in the sediments because they're dry. So now we have a big problem with asthma. So here is asthma is #1 disease, and it's because of the air quality. That is related with the water quality, right? Like it comes from the water. So yes, so I'm gonna do right now, I'm working with drones. So in the drones, I have like this machine. This machine, what it does is like take microorganisms from the air. So we're just gonna use it to see, to arrive to achieve different areas of the Salton Sea. Yes, so I'm going to field work in a spring break. That my spring break starts next week. So I'm gonna go to field work so I can take some samples of the quality of the air. I'm gonna looking for microorganisms, and I'm also gonna looking for the New River. I'm gonna go to Mexico because, as I mentioned, I'm from here. My family lives in Mexicali and Tijuana, so I go often to Mexico. I cross often. Last night, for example, I crossed to Mexicali just to eat because I miss my Mexican food, of course. And so just like go there, take samples in Mexico, and then just go back to US and then just take a look to all the distribution of the river all over the Salton Sea. This is the Salton Sea. So you see, it is apocalyptic. I've been there several times, and every time that I go there, I think a zombie's gonna come from the water and just take me out. Like it is really bad, the situation. In the 60s, a lot of people used to go there, like used to went, like travel, stay. There was like the Beatles, there was Marilyn Monroe, there was several like famous people visiting the Salton Sea. It was really famous. Now it's just a desert. There's nothing. There's some migratory birds sometimes, but not all the time. So we are losing it because of the poor quality of the water there. Okay, so my next destination is gonna be Antarctica. So I'm going this year, at the end of the year, in November I'm going. So I'm traveling from Argentina to Antarctica. So we were selected 100 women from all over the world, and yeah. So I belong to the group Cuarteto Antártico, that we are 4 Mexicans. We're really excited to be there. It's a really big deal. Usually Latinas, we are underrepresented in these things, so it's really important that we're there as a Mexicans, as a Latin America. There's going to be another colleague from Chile. There's going to be another colleague from Peru. So we are really proud to be this group of Latinas that represent them in this big expedition in Antarctica. Well, maybe I'm gonna just put a little bit of the video. Can you hear the video? I'm just gonna. Do you hear it?
No.
No, okay. I think I'm gonna have to reshare. Just 1 second, please. Share sound. Okay, there. Now you can hear it, right?
Yes.
Okay, better.
There are many things that concern people nowadays about what is happening to the planet. At some level, whether it's climate crisis or poverty, education or gender, biodiversity loss or the warming of our oceans, somewhere along the line we have to say, what kind of leadership got us to where we are today? And if we can understand that, ask ourselves, what kind of leadership will move us into tomorrow. Mother Nature really does need her daughters. More of us standing up to be collaborative, inclusive, legacy-minded. Homeward Bound was founded to create a global network of women with a STEM background willing and able to lead and solve our planet's greatest challenges. Right now, we're incredibly proud to welcome our 6th cohort, joining a global collaboration of 500 women strong, all chosen because they have the credentials and, most importantly, the will to work together to a more inclusive, legacy-minded world. The global COVID pandemic has given all of us pause to rethink what leadership at its best looks like. People have risen up, men and women, recognising the urgency of the crisis, working together, listening carefully to the people and how to enable swift, agile responses. These things will stand beside us as we move forward into what may be one of the hardest decades of the human experience. While the pandemic has given the opportunity for some of the best leadership practices to rise to the top, it has also created immense division through some of the worst practices. Greed and self-aggrandizement replaced collaboration and a care for the legacy we leave. That choice is no longer ahead for our species. If we continue down that path, it is an end of days game. Homeward Bound has an alternative. Homeward Bound focuses on building the skills of leadership, not just for individuals, but for the collective of the women of Homeward Bound. Personal, deep-level self.
Okay, we're just gonna leave it there. But that's the kind of the program. It's about like women in STEM, leadership, climate change, and we're going to Antarctica this year. It was delayed for 2 years because of the pandemic, so we're really excited to finally be able to go. Okay, so we're gonna continue. What are the contributions of my work to the society? Well, I would say like preventing like diseases, like cancer biomarkers, metabolite of toxin, and also emerging diseases related to the climate change. So as you notice, and as the video mentioned, like climate change is like a big deal, a reality, and we are like seeing diseases that we never saw before because of this situation, because of the climate change. So we're trying to predict this and be ready for the next pandemics. So we need to be careful about it. My career obstacles and how I overcame them. When as I mentioned, during my PhD, I have to study my third language, is French. So yeah, so my PhD was in French and English. It was very complicated because in that moment I never thought like learning another language, you know, like so it was a big deal. It was complicated. I'm not gonna lie. French is a really difficult language, but at the end, I mean, it gave me like another, I don't know, like another perspective of things, and another opportunity to see other things and talk with other people. Because sometimes I will recommend you to learn several languages because people opens better when you can speak their own language, and that's a reality, right? When someone that we talk and someone speaks Spanish, we open easily. So it is really important. Also, as I mentioned, my partner, he's French, and I have to talk French with his family. So it was, I mean, it is nice. I love to, and every time that I have meetings, and for example, I have a meeting and I hear that someone has an accent of French, I just start immediately talking with that person in French, and they think that it's wonderful that you can make an effort to speak another language. Mostly if you came from the from the US, because like there's a big reputation that Americans don't speak several languages, so they can really impress if I say I came from San Diego State University and I speak French. So they are really like, wow, oh, you do actually know another language. So it is really important that you are able to communicate with other people. Yeah, inequality in the classrooms. Gender discrimination also, like, you know, like not all the time you're gonna have professors who are awareness of women in STEM. So sometimes there's gender discrimination comments, you know, like there's comments, inappropriate comments from professors that they don't see that we are changing, the world is changing, and they will make comments like, what are you doing here? Like you're supposed to be in the kitchen, comments like that all the time. So you just have to let it go. You know, like, of course, complain, because that's not okay, that's not acceptable, but you're gonna hear this often. My sister's just engineering in biochemist, and yes, there was a colleague from her that all the time she was saying like comments like, you're here just in the meantime you have a husband. What is that? Super inappropriate. But yeah, those kind of comments. So you just need to like let go of those comments, or sometimes it doesn't even worth it to fight with these people, like to stand up. I mean, of course, stand up for yourself, but not like a conflict, you know. My career experience and how I made the most of them. Well, I was really lucky to get my PI position in as an assistant professor in YSA at the beginning. That opened me the door to several things. Also, I earned my grant, as I mentioned. I had a scholarship, a complete scholarship to go to Canada. And of course, I didn't have to pay my semesters because of my grades. And I always took summer classes so I can just advance on my career quickly. My tools for your backpack. So the first one is determination. Never lose sight of your goals. Like all the time, keep it here, so you can just remember them. And so when you feel like you're failing or you're tired, just remember those goals and keep it in your mind that your goal is bigger than what you're facing right now. So relax. And I have to remind this all the time. Honestly, it's really hard. Determination is strong. It's like you have to take it every day, and you have to prepare yourself of keeping that goals in your mind all the time, so you can just go for it every day. Never stop learning. That's something that I really love. I never stop learning all the time. I'm learning new techniques, new things. I love to travel and just learn things from the culture, from the language, from everything. Now I have some Brazilian friends, so I'm trying to learn Portuguese. I also try to learn Italian. So I all the time trying to learn. I love languages, and I love also like other cultures and eating. I love eating a lot. So yeah, like that's really important. And you keep your feet on the ground. That's really important as a PI, that always questioning yourself. And yes, and you know, like this kind of scientists who were like arrogant and like impossible to reach out. Not anymore. Like we as a scientist, we really need to be in touch with the needs of everyone, because we're supposed to be here to save, not to save, but to protect nature, of course, because that's my job, like mostly protect nature so it will not affect humans. But I mean, to protect and to take care of the situation and take responsibles about the situation that we're facing every day. I mean, climate change situations, we need to face it. We need to do something about it. So that's your responsibility as a scientist. You have a big responsibility if you decide to be a scientist. And is that like to really care about people and really care about the situation that we're facing every day. Imagine like if scientists say, oh, COVID is here. Oh, doesn't matter. No, we really need to take care of it. Okay, so now we're gonna work in the activity. So we're gonna identify the toxins around us, and we're gonna just try to see how many ingredients we can detect and how many of them are toxic or not, or what do we think is toxic or not. So okay, I'm just gonna stop sharing because I need to see you all. Okay, so you have do you want to start with questions? Do we have questions before we do the activity?
We're gonna do the Q&A at the end, Linda,
Okay.
if it's okay.
Okay, perfect. Okay. So, so all of you have your products? Yes, can you show me? Perfect. Okay, great. Okay, so we can take a look to the ingredients. Okay, And can you just start like writing to them in the chat, please, like all ingredients that you can see. Yeah, what do you see? For example, I have here, let's see, almost a late. Talk, mica, silica, okay. What else? Dimeticon, yeah. Yeah, I have the same. Usually you have like active ingredients and then you have like extra ingredients or inactive ingredients. Like, can you just like go to active ingredients and take a look there? Okay. Good. Okay. Sulfoxynate, okay. Sulfo. Aluminium, okay. Aluminium, okay. Tritalamina, limonel, propylene glycol. Okay, great. Okay, perfect. Okay, so now we know how to identify them. So we know how to read them. Where are they? So you see here it's say active ingredients, and then it's like ingredients just like that, or inactive. This one we just have active ingredients. They don't share with us. I know, yes, they do have a big list. This is sunblock because you know California, really sunny all day long. Okay, perfect. Great. Okay, so I'm gonna share again. Okay, now sharing. Okay, so yes, we identify for example, we can have fragrance, right? Fragrance can be like really urticant. They can be astringents for the skin. We have sulfates. They are carcinogenic. One of those, the one that is 1,4-dioxane. So maybe if someone can identify if you have sulfates or not, take a look at that. It's dangerous. Hydantoin and DMDM is carcinogenic, and formaldehyde also. Well, it's formed with this, and the parabens that are linked to the breast cancer. So every day we're finding new chemical products from our products that they are cancerogens or endocrine disruptors. Like it is all the time we're taking a look. And if you see ingredients, for example, in the shampoo, it says like shampoo for curly hair, shampoo for straight hair, shampoo for voluminize the hair. I don't know, like different kind of things. And if you take a look to the ingredients, they're pretty much the same. So I don't know why I just marketing what it tells you that is for curly hair or is for straight hair is pure marketing. If you take a look to the ingredients, there's no difference at all. So how it can be exclusive for curly hair? And sometimes some products for curly hair, because as you can notice, I do have curly hair, they're more expensive and they don't do nothing. So we need to be really identify the toxic, first of all, like what is toxic for us, but also like to be open to question ourselves all the time what they want to sell us, because you know the companies lie all the time. So we need to be open to understand if it's really what they say or it's just publicity, it's just marketing. We really need to question ourselves all the time. So I'm going to present to you some slides that I work on my social media. And then I'm going to talk about these two products that I'm showing today. So let's take a look. So this is the toxic of the lipstick. So let's just go too quickly. Just gonna slow down. So yes. So these products and lipsticks are lead polluted. Like not because they want to, it's just because, you know, they use like a pigments that are contaminated or polluted with lead. So they can be causing neurotoxicity, reduction of the fertility, changes in the hormones, and it can be bioaccumulation. It could be a bioaccumulation of the lead in the human. So it can be the teeth and the bones. So what are the recommendations? Is all the time take a look to the ingredients. Always take, every time that you buy a product, you have to take a look to the ingredients. So just to be sure that they are safe. And I will say, like, there's more strict regulations in other countries. For example, like US, we don't have good regulations of the products, unfortunately. So I usually, what I do is just buy European products. Europe has the most regulations related to toxic. So I try to use those products. For example, this product, this one is from France. So I try to use like those kind of brands just because I know that they have more strict regulations about it. And some of them, for example, are even cheaper than the American brands. So just be aware of that. Yes, it's not gonna appear in the labels, but the labels, but you need to take a look. Yes, and just like try to find like more sustainable ways to use. Okay, so that's the first one. And then we have of the mascara. So yes, so we're gonna have paraben, phthalates, thimerosal, and all of these are really toxic. Also, like the dyes are really bad. Even in the food, dyes are really bad. They have been discovering every day, like red 31. They're carcinogenic. So we need to be careful about that. And formaldehyde. Formaldehyde is one of the worst. Recently, I don't know, have you read? I didn't show that one, but I can show you from my social media. But recently they found out that there's like the NIH, that is the American Group for National Institute of Health in here in the US, that there is a correlation between the products to straighten the hair, you know, the straightened products related to cancer. So there was like a lot of Latinas community and African American communities, they were really affected by this because, you know, we have curly hair, we have wavy hair, you know, so we use a lot of products related to that. And it was formaldehyde, one of those who are really dangerous. So yeah, so you have to check all the ingredients all the time. As I mentioned, change how frequently you buy your mascara. For example, they recommend 3 months because you do have microorganisms there, so you need to change it, and just to find like more clean options for you. You can make your own mascara really easily. Actually, you can just need charcoal, and you just need like what is the name of that, aloe vera, and that's it, the gel. Okay. The next one is related to the hair. So yes, if you dye your hair, just be careful because it can have ammoniac, that it can let you blind. Ethylamide, peroxide, of course, phenylethylamine. So just try to use the products, just to use the products that they say that they're not toxic or less toxic, or they don't have these compounds. But I can stop it. Okay, and this is the sunscreen. There's two kinds. This is the chemical sunscreen, and this is the physical sunscreen. So you can take a look to you. If you have a sunscreen, just take a look which ingredients you have. So if it has like oxybenzone, homosalate, or those kind of compounds, that one is a chemical. It means that you're gonna absorb the chemical sunscreen. But if you use a physical sunscreen, it means that you're not gonna absorb anything. It's just gonna stay there. Honestly, it's kind of annoying because it's white, so you're gonna look like you have a white thing there, but it's more healthy. So sometimes we need to balance what we prefer, and we've been avoiding to make this question all the time. We just think, oh no, I just want to look good and I don't care if it's healthy or not. Well, we're gonna have repercussions in the future, so we really need to be aware of these things. So if you take a look and you have titanium dioxide, dioxide and zinc oxide. In that case, those ones are going to be the physical sunscreens. But that's it. So if you want to follow me on my social media, I'm going to be publishing some pictures of the Antarctica when I'm traveling there. And yes, so thank you so much for your attention. Just gonna stop sharing.
Thank you, Linda. That was amazing. Now we're gonna do the Kahoot. So I invite Karen. She will lead this part of the session, so you guys be aware to participate. Karen, you have the floor.
Okay, can everyone hear me? Just to double check if my hearing is correct, because I was having issues. All good? Okay, so I am going to be hosting the Kahoot for today. So if everyone could just log in, the pin is there in case you can't see. I'm just going to quickly write it down in the chat. Okay. Okay, we're starting with the first question. Question, what areas are included in the eco-toxicology? Would it be ecology, toxicology, and genomics? Would it be biology, physics, and mathematics, biology, toxicology, and physics, or ecology, biology, and physics? And the first one would be read answer. So congratulations to all that got it correct. So while we're waiting for the second question, the question is, in central ideology of microbiology, the correct order the molecules would it be RNA, protein, and DNA, protein, RNA, and DNA, or DNA, protein, RNA? Great. The answer was the yellow, so congrats to the team that got it. And we would move to the third question. Third question. Oh, wait. Sarah's in the lead. Congrats, Sarah. Third question. What is the meaning of paroscindemic? Is it to cure cancer, boost the growth of cancer cells, that it cures cancer, or is it heterogenic? The answer was green. It does cause cancer. Not just don't confuse itself with the growth of cancer cells. So in the lead, we have Anna. Congratulations. Second question, what type of mutagen is radiation? Is it chemical, physical, biological, or hormonal? And the answer is physical. See, there was some sort of confusion there, but it's okay. It's a learning process at the end of the day. And last question, what type of mutagen is oil? Is it physical, biological, or manufactured? And the answer is, it is a chemical. So that's, even if you did get it wrong, it's a learning process. Don't let yourself down. And let's see who our winner for this game is. We have Linda. Let's see who is in your lead. So congratulations, Louis. You won. You're all winners at the end of the day. So just kind of it will be next time, okay? Congrats to our winners.
Great, Karen. Thank you very much for being in charge of the Kahoot, and congratulations for number one and for everybody who participated. You did great. So now we're going to start the Q&A. Please remember to raise your hands or to write them directly on the chat, and I will read them. Take the chance to ask all the questions you had during the presentation. Yes, Karen? Do you have a question?
Yes. I just wanted to ask in regard to dyeing of hair. Of course, it is depending on the chemical. So are there also chemicals that can refuse to dye on your hair? Like it wouldn't have an effect. Does that also cause
I'm sorry, Karen, it cuts a little bit. It cuts in the question. Can you repeat it? I'm so sorry. Please.
Oh, okay.
Or if not, write. You can write it, Karen.
Nah, let me write it instead.
Sorry, it's my internet. As I mentioned, it's failing a lot. I'm so sorry.
I understand.
While Karen writes the question, we have another one. What are you doing in Antarctica, Linda? If you can specify.
Okay, I'm gonna do like a visualization of the women in STEM, and also like for the climate change, we're gonna take pictures and observe the effects of the climate change in that region. The region is like it's like a kind of a big lab from the world, so we just want to see like the effects of the climate change in this region. So that's my main purpose, is related to climate change.
Great. How long will you be there?
1 month. It's an expedition for 1 month.
Great, great. We have, well, I see Karen did not write it, so Alejandro, do you have a question?
Hi, you listen to me, right?
Yes.
Nice. First of all, it was amazing, your exposition. I love it. And I want to ask about like environmental toxicology. I find, as you have mentioned, the impact that toxins have in the environment are very harmful. So like different ways like young people like me can do to change this. What recommendations can you give to us?
Yes, like to the environmental health, like environmental toxicology. Yes, I would say like you can start like I remember when I started as a little girl as my own ecological club. I mean, it was only me and my family, but you can always like start I don't know, like taking a small groups of friends and just trying to do something like a problematic that you are facing right now in your community, and maybe just trying to do something about it. Like I don't know, when I was living close to the thermoelectric, and they were like they didn't have like a filter, so they were polluted. Every morning you wake up and you will have issues, like a lot of issues around our cars, and that was directly to our lungs. So yes, I started like this club so we can ask for demands to the government, or just like express our concern related to the H's released by the thermoelectric. So it is kind of like that. You can start with the small things, or maybe just like open like a place that you can recycle like things you can collect. I mean, it can start like a small things, but believe me, there's no small things in ecology. There's no small things in climate change. We can start like being more responsible about consuming what we consume. Maybe tell to our parents like, hey, you know, like maybe take our own bags instead of like using plastic bags. When you go to a party or you have like party with your friends, don't use plastics. I didn't talk about my projects related to microplastics, but I'm working on that too. They're really dangerous. We're never going to eliminate those ones from the environment. So yes, maybe start with small things and try to consume less. Try to, you know, try to be responsible about what we produce, because every time that we consume something, we don't eliminate, right? And there's try to find like other ways so you can buy more responsible things. So if we demand responsible things, the companies are going to produce them. Right now, nobody demands it, so there's no lot. If you take a look to the market, how many of the market really it's like really ecological? Not a lot. So we need to start demanding those things. And also you can prepare your own things. Take a look to the recipes online. I'm working in a book right now talking about that, how to prepare your own deodorant, how to prepare your own mascara, how to prepare your own lipsticks, and all this ecological. You know, like for example, my nail polish is ecological. So you can prepare your own things in a safe way, and you will be safe, and also you will spend less money, and also you will be responsible with the environment. I hope that responded the question, Alejandra.
Yes, thanks. Thank you so much.
Thank you.
Yeah, so sorry, Alejandra. I don't have my glasses and I read Alejandro, but now I see it's Alejandra. We have the question that Karen wanted to do. Linda, in regards to dyeing hair, is there any certain ingredients that do not work on everybody? And she wants to know if it is necessary to bleach the hair first and then dye it. What do you would dye it, or what would you recommend?
Yes. Okay, Karen, great question. Yes. In regards to dyeing hair, can your hair not dye because of certain ingredients? Yes, it can happen. For example, that some ingredients cannot be absorbed, or they cannot take it. They cannot be penetrated in the hair. That's a reality. And if you bleach it is peroxide. So it's really aggressive to the hair. And if you have like, for example, I cannot bleach my hair because I have curly hair. If you have curly hair or just wavy hair and you bleach it, you will destroy the protein in the curly and it will make straight the hair. So I did so many mistakes when I was young, dyeing my hair. I did several colors, and now I have short hair because I have to cut it completely, but because it was destroyed. So I would recommend you like maybe take a look to the henna. Henna is a really good, it's a really good product. It doesn't damage the hair. It doesn't contaminate at all. So henna is really good, and you can mix hennas. That's something that I didn't know, and I have a friend who is super good on that, and she's mixing hennas. You know, like she's mixing red with the brunettes, and it makes beautiful color of hair. You know, kind of a Cooper like color. I love it. And yes, you can always make your own mix. I will say I love to experiment a lot, so just go for it. Try this henna and maybe try in a little hair, and if you see like it's good for you and it's working and it's penetrated, go for it. Otherwise, just take a look. Obviously, take the precautions and read all the precautions online, because your products will have like, you need to do this, Don't let it to touch your skin because it will stain your skin. It's super good to stain everything. And my recommendation is to make like a lot of use like a lot of protection, you know, like towels around, because it's gonna be a mess no matter what. It's a messy product, but it's ecological. It's not harmful for the health. Just be careful. But yeah, I hope that answered your question. Okay,
Yeah,
because I dyed my hair. It's curly, 2 different products, and it didn't work. Yeah, maybe it's the porosity of the hair. You know, some dyes doesn't work in my hair. I have a 3A hair, curly hair pattern, and it sometimes not all the products works for me. Henna is really strong. I mean, strong in a safe way because it's a natural dye. So my recommendation is you can use the henna and put it like you know, like this kind of hats for the hair. Leave it there for hours. Henna, for example, I use it 3 hours. I leave it 3 hours in the hair, and it works for me. I mean, just make an experiment and let's see if it works. But there's no like in henna we don't have like a lot of options. There's only like black, red, brown. I think it's blonde, and you can mix with all those 4 colors, and you can just make your own, the one that you like. I love Cooper. Maybe you can try Cooper if you're interested. I don't know which color you were looking for, but Cooper is beautiful, and you can try it if you like it. You have that option, ecological and safe for you.
great, thank you. Elvia, you have the floor. Ask the question.
Well, first is thank you for the class. It was, wow, I loved. And I have some questions. First is that how does Niño de la Tierra looks like, or what does it do, you know? Because I see, watch that it is like a little insect.
Yes. Okay. Yeah. Yes. Thank you, Elvia. Yes, it's like an insect this size, maybe. Yeah, not so big. And it looks like it's weird. It looks like it has a face and it looks like a kid. Actually, it doesn't look like a baby, but it's weird. It has eyes and, you know, like it seems like a nose. But yeah, it looks like that. I can take a look. Let me see if I can share a picture of that. Look. Look, I'm just gonna share my screen so you can take a look to this one. Maybe they don't call it like that. I don't know. Do we call it like that in California? Like this is, you see, it has the eyes. It kind of look weird. Like I don't know. They call it Niño de la Tierra. Honestly, I don't know why. But it's like that. It's a little insect. And what it does, the toxic part, is just like if you touch it, the skin release like a kind of astringent thing. So that is the danger, is that you can absorb a little bit, because you don't absorb a lot in the palm, but you can absorb a little bit and be toxic for you. But it's nothing deadly. You're not gonna die or something like that. I mean, it's just a little bit toxic, but you have to be precautious. But yeah, that's the Niño de la Tierra.
Okay, thank you. And all kinds of lipsticks, mascara, and sunscreen are toxic, or just the one that has a specific ingredient?
We always have to double check, because there's different companies and different products that, and there's some products that are being released as not toxic. You always need to check the ingredients. What my recommendation is, like, take a list of your products, of your ingredients, I'm sorry, and just looking for online. There is the website of the IARC. It's the French Institute for Cancer, that is from the United Nations. It's in Lyon, France, and those are the ones who release the handbooks to say which products or which chemical products are toxic and cancerogenic or not. So just my recommendation is always take a look to the list of those products, because sometimes it's difficult. You know, I don't know, have you listened that that new concept that it calls greenwash. Greenwash, it means that they tell you that it's ecological, but actually it's not. So we have a lot of greenwash lately, you know, like, oh no, because they know it's a fashion.